syllabus in case anybody's a little late. We usually will be covering any lecture material outside of the class in way of videos, so you don't have in case there's my echo, so you don't have too much homework outside of class. So we'll be doing lecture outside and then any work in here while I'm around to answer questions, but didn't want to give you anything to watch for today, so I will talk for a little today. Okay. I am gonna go ahead and share my screen with you all as we run through a couple things. Okay, so yeah, welcome to Bio of the Cell. I'm Dr. Zelazny. I've been at the college for about 2 years. I'm an anatomist by training and a paleoanthropologist. So I study the structure and development of bones in the course of human evolution. So I also teach A&P, as that explanation might suggest. So I hope I'll see many of you next year. But I've also been teaching Bio of the Cell for many semesters now. It's a short course, so it feels like I'm always teaching this. I'm really excited about it. I know this class has a reputation for being a fair bit of work, and it is fast. So that is true. So it will prepare you well for A&P next year in terms of how much you should be studying and how much you should be working at this class. But don't want you to freak out too much. We have a lot of support and resources for this class, and we have a lot of time physically in class together to work through the material, and most people end up doing pretty well in the end. So if you had any nerves about this class ahead of time, I hope that through the course of the class you'll ease into it, and we'll ease into it today too. Okay. Speaking of support and resources, we have a great SI instructor who I don't believe is with us right now. Let me just check. Brooklyn, you're not here right now, are you? Okay. She's not with us right now, but she shared this slide with me. So Brooklyn is our SI leader, and she's been doing this for about 2 years now as well. So SI is a great place to review class material and ask any questions that you didn't get a chance to ask in class, or hope you guys won't be nervous of me, but if you were too nervous to ask in class, Brooklyn is a great resource. So SI this semester is held in the evening on Tuesdays from 5 to 7, or in the morning on Thursdays from 8 to 10. Brooklyn's email is also up on these slides, and I'll make sure to share this set of slides with you at the end of our class period today so that you can reach her. But there's also an SI tab linked in our Brightspace course. So if you're not able to attend SI at these times, any materials that Brooklyn goes over with people will still be up. You won't get the opportunity to sort of have a talk back and forth with Brooklyn, but the practice can be helpful as well, although we will do a lot of practice of things in class. So the worksheets aren't mandatory, SI is not mandatory, but I've heard from people in previous semesters that it's really valuable to the students who do go, and the students who do go feel much better prepared as we approach our exams for this class. So if you are able, I recommend at least popping in. Okay. So Brooklyn will also share with you a folder with the answer keys to those worksheets, again, in case you're not able to make these times. Okay. So I want to talk a little bit about the general structure of the course before we get into things. So the daily structure is going to work something like this. Before class, every day but today, you will have a video assignment. So basically a YouTube video that you will watch. It'll be my face talking at you through some material. There's a set of Google Slides, a folder of them linked from Brightspace as well, so you can follow along. There are readings listed along with those videos in our course schedule. Those readings for this semester, I would consider supplemental. So if there's something in the videos or in our listed learning objectives, which I'll talk about in a second, that you don't understand or you still feel unsure about, I have the page numbers and sections listed out for you there. Our textbook is free. It's available online. You can download a PDF or you can view it directly in your browser. So as I said in my email yesterday, don't worry that there was something you need to buy. I like to make this course as cheap as possible for people. But it's still a pretty good textbook, and it really does have everything you need in there if you are confused and want to go a little deeper. Okay, so you're going to watch those videos and check out the readings if you're confused or you feel like there are gaps in what you want to understand. You'll have an individual quiz on Brightspace to go along with those that you will be completing before class. And when I say quiz, this is pretty low stakes. This is not something you need to be studying for or panicking about. This is just to sort of guide you along the videos and guide you in your thinking on the learning objectives for that day and the readings. So it's untimed. You have between the time we end our scheduled class time and the next time we meet, you just need to make sure that you take a look at that and you get it done before we meet. So you will have a quiz on chemistry before we meet next time on Wednesday. So that'll open up at the end of the class period. You can do it on Tuesday, you can do it on Wednesday morning, just get it done before class. Because we will spend our time in class working in teams on a group quiz and on problem sets. So every class period is going to start out with about 10 minutes for our team quiz. And here's why you want to be extra sure you do the individual quiz before class, other than you don't want to get a 0 on easy class assignments. Those team quizzes are going to be the same questions. So the answers won't be available until after the group quiz. So at that point, you'll have a chance to discuss what everybody wrote down as their answers, discuss any of the material with your team, sort of finalize your answers or take a second chance after you've talked through the topics with your classmates. And the reason for that is that a lot of studies show that actually talking through these things with people who are also learning the material can really help to solidify your understanding, and just talking about concepts really helps you learn stuff better. So again, the group quizzes, not meant to be stressful, should be an opportunity to earn some easy points together. After that, we'll pause to ask questions. So you'll have watched the videos of me giving lectures before class, but I still want to make sure that you have a chance to ask questions, anything you're confused about, go over again. So we'll pause to do that before we move on with our material. And the bulk of class is going to be doing a team activity, which is sometimes going to be a case study. So we might have an example of some type of disease that's related to cellular biology, and you'll work through it using the knowledge that we're learning as we go forward. Or it might be a problem set with some practice questions. You might have to define terms. The idea of these is to get you thinking and get you working with this material, because that's a better way to practice and a better way to study when you go to study on your own than just sort of eyes glazing reading for hours. Okay. So we'll be doing those in teams because they're harder to do on your own. And again, practicing explaining things to your group mates and hearing people explain things like 5 different ways when you have 5 different people in a group is really helpful for reinforcing your learning. So that's what we'll be doing. And at the end of the week, you'll have a short discussion post or a survey after class. It's not going to be super structured. It's not that kind where you have to post it and then respond to 3 people. I personally find those a little exhausting. The point of the discussion posts is to do a little reflection on how the week went. I will want to know anything you're still confused about so that I can kind of touch back on that in class. Might ask you how things with your groups are going. I do pop through frequently to talk to groups as they're working, but don't have eyes on everybody at all times, and I don't want you to get stuck in an uncomfortable situation with your classmates. So those are great times to let me know that. And again, chances to get easy points. Okay. So mentioned the learning objectives as a way that you should be guiding your studying before class. So what you're writing down as you're taking notes. And then your learning objectives are also basically going to act as a study guide for later, for when you're studying for the exam. So I'm going to actually click out of this and get us to our set of learning objectives. So our learning objectives look like this. They are lengthy, but they are literally everything you need to know for this class. So today we're going to talk about something called cell theory. We're going to talk about the difference between prokaryotic and eukaryotic cells. So that's what's on your learning objectives for today. So when you're studying, this is just your reminder for exactly what we did for today. They're also structured basically as prompts. So if you were feeling really gung-ho, right, you could write down, pretend this is an essay question, actually define this 3 tenets of cell theory, right? So you'd give that to yourself as an essay question, and then write on your own how you would define them and check your answers against the slides, against the readings, and so on. So that's how you'll be using them for studying and for kind of preparation. And they're broken out basically by topic or by day. So this is what we'll be doing today. This is what we'll be doing Wednesday. This biological macromolecules thing is really big. We'll be covering that over 2 days and so on. Okay, so back to our learning objectives. Okay. So you're going to take notes on these. You're going to organize your notes by these. You're going to reorganize your notes by these, and you're going to quiz yourselves on these. And hopefully that means when we get to the exam, you will be able to do these things. So they are your study guide. I often get questions about study guides. Literally, these learning objectives are a huge list of potential essay prompts, ways you can form questions for yourselves. So those are our study guide for this course. Okay. So I said that we're going to begin every class with a group quiz. This is not strictly a quiz. I figured we'd take a little time to pretend we're doing our normal schedule. So we're going to start today by filling out the course survey. You'll get points just for doing this. So I'm going to pop this link in the chat, but it is also listed under the content for today. Let me find where our chat went. There we go. There's our chat window. So here is our link in the chat. Okay, and the other place you can find this is linked. If you go to our homepage, go to content, you'll see the first thing we have in week 1 is this introduction survey. I'm the only person who can see your answers, so don't feel nervous or self-conscious about anything you write. But we're going to take a couple minutes to fill this out so I can get to know you better and we can make sure that we're all on track to start this class. Okay, looking good. Take another minute or so to finish those up, and we'll get back to it. Okay, I think I have almost everybody's responses now. Awesome. And I'll go through and make sure you guys get points for that after class. Already starting out with 100%, right? If you turn that in, doing great. Okay. So the next thing I want to do is just make sure you know what's up and coming. So in Brightspace, everything is organized by week. So all our week 1 materials are under the week 1 tab. But these are some upcoming assignments we have. So hopefully everybody got in their introduction survey just now. Gave a little wiggle room for anybody who wasn't able to make it to this class today because they forgot today was the first day of the spring 2 semester. I know our schedule can be confusing. And so at the end of class today, we'll also be doing a short kind of introduction practice problem set. So you should have enough time to turn those in by the end of class. And again, you'll get credit for doing those. Okay. For next time, you'll have some videos to watch. And as you are doing those, you'll be filling out your individual quiz. In class, we'll then take the same quiz again as a group. We'll do a problem set on chemical foundations. And then for Friday, we'll be going over carbohydrates and lipids. So same deal, you'll have a video to watch, we'll do an individual quiz, and then in class, we'll do a group quiz and a problem set. Okay. So I find having the professor read the syllabus out loud is not terribly productive for everybody. And also I feel really weird reading a syllabus out to you guys. So we're gonna try something that my friends in tech companies told me they've been doing. So we're gonna have a short silent meeting about the syllabus. So I'm gonna link you directly to this copy of our syllabus. And what you're gonna do is you're going to pull it up in your own, and you're gonna read through it. And as you have questions, you're going to use the commenting tool to put them in the comments. So I'm going to show you what that might look like. So here, oh, perfect. See, a bunch of you have gotten in here, making sure I'm in the right one. Okay, so here at the top, you'll see this commenting tool. Oh, there you go. So you could add a comment like this. If you have a question, just add a comment here, and in a couple minutes, I'll skim through, and anything we have questions about outstanding, I'll make sure that we go over as a class. Okay, so take a few minutes to skim through this. You'll note a lot of this is links. So this is just your link to the textbook so that you have it there. You'll see that we have links to some study tips. And we have links to where you would be able to find tutoring schedules if you decide that you want to meet with a tutor about this. We also have links out to our schedule, including the video links that we have for this week. Okay. So I'm going to be quiet and give you a second to read through this. Okay, I'm gonna launch a quick poll. Just checking if everybody's made it through yet, since I'm not seeing a lot of comments, but hopefully that just means things are clear. Okay. Seeing lots of yeses, which is awesome. So let's go ahead and take a quick look through. So you have all the information about how to find me, how to find Brooklyn, a little bit about what we'll go over in class, some tips about what you should be doing both in class and outside of class. Here we have our assessment, which I know is one of the things everybody goes to first, which is why I highlighted it nice and convenient for you. Okay, so Sydney asked if there is a final exam. We will be taking an exam during finals period, but it's not a final exam per se. Our final exam is just going to be this exam 2. So it's not actually cumulative, although all our material does build on itself throughout this course. But this will be focused on the second half of the material, and it'll be worth 25% of your grade, just like the first exam. So that is our final for this class. Okay, let's see if we have anything else. Not seeing anything. Okay, so again, I'll keep updating this tentative schedule. I don't want us to get too far ahead of ourselves in case we have any stumbling blocks in class, but this is all updated with our general topics and with our videos through for all the way through Monday. So if you know already that you have some things coming up this week, go ahead and you can get ahead if you want. Okay, so I'm going to pull us back to our introduction in that case. Okay, so we're doing a silent meeting. Okay, so there's one thing I want to talk about before we get into the meat of the material today. So I wanted you to take a guess at what some of these discoveries have in common. And looking for my chat, having meeting controls up and Google controls up at the same time is pretty tricky. Where are we? Okay. So if you have any guesses, go ahead and make a guess in the chat after reading through these. Okay, so I'm getting a couple answers that all are studies of cells, which is absolutely true. They all actually also have something else in common, which is not as obvious and is a slightly more uncomfortable topic to talk about. So this is not a history of science class, which I'm very grateful for, because these are not topics I'm super used to discussing, but it is important as we start our study of cells to realize that the history of how some things have been discovered can be a little icky if you dive deep into it. And as I said, I'm not very practiced at having these types of discussions, but I'm trying to do better for myself. So if anything I say makes you uncomfortable or you're not in the same place as me, feel real free to have your own emotions and share that back with me. But these are actually all discoveries that came from the cells that were taken from this woman called Henrietta Lacks, which you may have heard of before. So she died in 1951 of an aggressive cervical cancer. Now, her cells were initially taken to study that cervical cancer, but without her knowledge or her family's knowledge or anybody's permission, they were actually used for tons and tons of discoveries, and they still are used to this day. And in biology in general, if we look into the past and how biological research was done, we run into a lot of thorny issues like this. So like I said, we're not going to talk about these much through this course. We're going to focus on our knowledge of cells. But before we get started, I do want to make sure that we appreciate a little bit about where some of this knowledge came from. And so Henrietta's cells were taken by these doctors at Johns Hopkins Hospital, which is actually where I did my PhD. So I feel a little uncomfortably close to this. And we still use her cells for research to this day. So her family never got acknowledged in this process, and they really want people to know about who she was as a person. And she loved to dance and cook, that she was super generous and the center of her family. And even they, acknowledge that her cells have done good things in the world. So they're not trying to cancel all research, but it's important that we know that things like this aren't okay as we move forward in biological research. And it's super important that as we move forward, everything is really done with informed consent, because biological research has not always been the best about that. There are other communities that have been impacted by this as well. So there was a lawsuit, which for me doesn't feel that long ago, 2004, but for you guys, I'm sure it feels like almost prehistory. So members of the Havasupai tribe donated samples in 1989 for diabetes research to help their community. But after that, these cells were used without their permission to study a lot of much more controversial topics that the tribe was really not okay with, like schizophrenia, population inbreeding, ethnic migration, really, really taboo topics for them and generally at the time. And there's a history of scientists using samples that were taken from people without their permission to kind of prove their inferiority. So there are a lot of people to this day who really don't want to give up or give out or share information about their biology. You may have heard some discussions like this with things like 23andMe, right? Or these genome projects that are trying to find criminals. People can feel very, private about these things. And some of that has to do with the really damaging history of some of the ways that research has been done. So I realize that you are all college students, but in science, sometimes I've heard some scientists get a little bit, offended by talking about these ideas and thinking that how could people not want to participate and stuff like that. So as you go out in the world, knowing things about biology is super cool, but if you encounter people who are uncomfortable with some of the stuff you're learning or some of the ways it was learned, there's good reason for that. So I do want us to be aware of that, because we'll be talking about some topics that are general interests like genetics and stuff, and it sometimes makes people uncomfortable. So you'll also have noticed in the syllabus that there is a land acknowledgement at the end. So I do want to acknowledge that whether you're joining virtually or from Duluth, we are all living on the lands of many Indigenous peoples. So we do continue to benefit from historical damage, right, that was done to people. Specifically in the case of Saint Scholastica, the Anishinaabe and Sioux people were the original people and caretakers of this land, and we benefit from this land now, and they didn't give it to us on their own, much in the same way that biological research has misused a lot of biological data. So that is my disclaimer at the beginning here, and I think it's important to talk about, especially in the times that we're living in. This is a lot of sort of conflict in the public sphere, and I just want you to be aware of how this relates to biology as we get started out. Okay, so that said, we are going to dive into our first topic on cells, which is prokaryotes versus eukaryotes. So this part of our slide set is available to you already in our slides folder. So if you want to open that up, I will give you a second to do so. But again, this will be on video. So if you want to touch back and take notes after we talk, that's fine too. Okay. So prokaryotes and eukaryotes is a very simple division of different types of cells. So before we even talk about their differences, I want to make sure that we know basically what a cell is, right? So cell theory is basically talking about our fundamental understanding of what cells are and where they come from. So we consider cells the basic unit of life. In order to be considered alive, an organism needs to have cells. That could be one cell, could be 3 cells, could be 1,000,000 cells. At least one cell, that is the unit of life. Second tenet, so second rule, tenet means rule, of cell theory says that all living organisms are made of cells. So that's like the sort of thing that goes along with understanding that cells are the unit of life, right? Cells are the unit of life. So living organisms, they're made of cells. All of them are made of cells. It's not like cells are alive and living organisms are made of something else. So cells are the unit of life. Living organisms are made of cells. Number 3, cells don't come from nowhere. Cells come from pre-existing cells. So in our second unit, we'll be talking about cell division. So how cells split, make new cells, which is part of how living organisms grow. So now, different types of cells. Our big main division between cells is going to be between something called prokaryotes, which are going to be really simple and small, and eukaryotes, which are going to be much more complicated. They're going to be the type of cell we see in like humans and multicellular organisms, and they're really going to be the focus of our course. But because this is also a prerequisite course for microbiology, I want to make sure we also know what a prokaryote is. So cells are all, prokaryotes and eukaryotes, going to have some things in common. So cells are all going to have a plasma membrane. So plasma membrane, and I'll pull up my pointer tool. There we go. So the plasma membrane surrounds the outside of the cell. And we're going to talk about the plasma membrane in deep detail next week. We're going to be looking deeply at its chemistry, which is why we have to cover a little chemistry review on Wednesday. Inside every cell, prokaryote and eukaryote, we have the cytoplasm. So this is going to be fluid, proteins, very cellular components, this kind of gel, liquid stuff floating inside the cell. Every cell is going to have DNA. DNA is going to be contained in sort of different places, depending on whether we're talking about a prokaryote or a eukaryote, but they are going to have DNA, which contains genetic information. And finally, we're all going to have ribosomes. So prokaryotes and eukaryotes have ribosomes. Ribosomes make proteins, and proteins are kind of what makes the world go round, as you'll see throughout this course. Okay. So Our two kind of cells, our prokaryotes and our eukaryotes, might look something like this. So here we have a prokaryote on the left, and I believe this is an image of E. coli. And here we have a typical animal cell, which is an example of a eukaryote on the right. You can immediately see part of what I mean about eukaryotes being much more complicated, right? You can see all these different colors, these swirling internal membranes, these little pockets in the eukaryote. So that's going to be one of our differences between prokaryotes and eukaryotes, but we're going to say it a little more scientifically. So prokaryotes, those simple cells like bacteria, although there are prokaryotes that are not bacteria, prokaryotes lack a nucleus. So I'm going to flip back to that previous slide with the picture for a sec. So prokaryotes don't have a membrane surrounding their genetic information. That's where the nucleus is. So in red in both images, we have DNA. But in the prokaryote, it's just kind of floating in the center here. And in a eukaryote, it's contained within an internal membrane called the nucleus. Second difference is that prokaryotes lack organelles. So organelles are all those other internal membranes, right? So here in pink, we see our endoplasmic reticulum. In blue, we see the Golgi body. In orange, we see mitochondria. We'll talk about all these organelles later. Prokaryotes don't have them. Number three, prokaryotes are much smaller than our eukaryotic cells. So here we actually see a size comparison of a virus versus a prokaryote versus a eukaryote. So if you look here, the prokaryotic cells on this image on the right are here in the darker color. The eukaryotic cell, you can see way, way, way bigger than those prokaryotic cells. And if you tried to imagine what a virus would look like next to the eukaryotic cell, crazy small, right? So another thing to know is that all prokaryotes are unicellular, so one-celled organisms. In prokaryotic DNA, instead of being in structures called chromosomes, we'll talk about chromosomes in Unit 2. Prokaryotic DNA is going to be in a sort of specialized circular chromosomal structure. Okay. So here are our two cells, again, the prokaryote and the eukaryote. Yeah. So viruses are not on your learning objectives, but given what's going on this year, I thought I'd make a note about viruses. So viruses do not count as cells. Viruses don't even really count as alive, which is why we call them acellular. We don't count them as alive because they really can't produce energy on their own. They don't all have DNA, and they can't reproduce on their own either. They need to use host cells. So like COVID-19 has to use your host cells in order to make more virus. Okay, so that's one of the big differences. So when we talk, we're not going to talk really about viruses in this course, and that's why. It's because this is a cell biology course. Viruses are just a little bit different. So they look kind of like this. Don't have organelles, don't have DNA, don't even all have an outer membrane, although some do. Okay. So what we are going to do now, I'm going to scroll through this. We're going to break out into groups, and we're going to do our cell bio in my career problem set. So this is based on a survey one of the instructors for this course did of her various Facebook friends who are now in scientific and healthcare careers about what they found most important from cell biology. So we're gonna break out and do a little practice on this. You're gonna practice copying a document, and you're gonna practice editing a document with a group. And at the end of the class period, we have like 15 minutes to do this. You'll upload this. I'm probably not going to have time to share out our responses with our commonalities with our group in class this time. So we'll save some team bonding for next time. So we'll just get started on this problem set. Okay. And again, this is just practice, so there are not strict right or wrong answers for today, or at least I won't be grading for correctness for this problem set. So I'm going to make sure that this link is quick in our chat, although again, it's linked on Brightspace in our tabs as well. And the instructions for how to do it are also on Brightspace. But basically, you're going to make a copy. You're going to share it with your group members. So you're going to add them using the share button on the right. And then you're going to edit it together as a group. Okay. So we'll just do randomized groups for today. And for Wednesday, I'll form some permanent groupings after taking a look at those surveys you sent me. Okay. Hey, Kevin, you still with us? Okay. I can assume you went to the bathroom or something for a second. You'll see a group invite when you get back.
Hello.
Hey, how's it going, Kevin? Do you see a group invite?
Huh?
You should see a group invite to a breakout room right now. We're working on the cell bio in my career problem.
Oh, sorry about that. Now I see it because mine's disconnected.
Yeah, do you see it now?
Yeah, no.
Okay.